Thanks, John, for the nice introduction and the invitation to come here and talk about some stuff that we've been doing over the last, 5 or slightly more than maybe 5 to 7 years or so, looking at the mathematics of wound healing, and I'll explain what I mean by that as I go through the talk. And this is the School of Mathematics in Bristol. That's the building, from one view anyway. So a view I particularly like because that's a Voronoi tessellation. So wound healing, well, unless you've had a very privileged background, you all have experience of having wounds and hopefully having them heal well. And if you want, typically when we think about wound healing, if you read a biology textbook or something like that, you'll hear that wound healing has 4 stages. First there's blood clotting, and then there's inflammation, or the immune system kicks in, and then there's tissue growth, some growth of tissue to close the wound hole, and then tissue growth is often called re-epithelialization. Epithelia is a word for an interface, a membrane. So essentially, it's the reformation of some barrier membrane protecting the internal bit of the body from the outside. And then finally, there's tissue remodeling. And these things are typically happening over scales of minutes to weeks, or if it's a very bad wound, maybe months. I am actually today only going to talk about this bit, the re-epithelialization, or this tissue growth bit. And I kind of want to just give you a sense of how, in some sense, how little we know about wound healing. And I'll say what I mean by how little we know about it, in the sense that I think what we know currently basically is rather qualitative, and really one would like it to become more quantitative, and of course that's where mathematics comes in. Okay, now it should be obvious that it's something which is clearly important for society, so therefore it's a worthwhile problem to study, though one shouldn't study mathematics just because it's worthwhile. One should study whatever is interesting. So hopefully the experience most of us have been what are called acute wounds. These are wounds that heal in a predictable way, and after usually maybe a week or 2 have healed. However, there are other types of wounds, and those are called chronic wounds, and those are wounds that don't heal, and sometimes can be years or even decades. And this is an example of a chronic wound, so not for the squeamish. And the NHS definition of a chronic wound is 3 months, a wound that hasn't healed after 3 months. So after a wound hasn't healed for 3 months, then you move from one part of the NHS to the different part, which is dealing with chronic wounds. And chronic wounds are a huge burden on the health system. The NHS spends a huge amount of money on chronic wounds. Now I'm going to ask questions during this talk. So this is my first question. How much do you think the NHS spends on chronic wounds a year? 1,000? 1,000? Mm-hmm. Higher. 1 million? Higher. Higher.
1 billion.
Higher. A little bit higher. Okay, I'll tell you, I think the NHS spends 8 billion a year on chronic wounds. Okay, so it's a really important part of the health system. So, and essentially, we don't know why are some wounds chronic, and the reason why we don't, one of the reasons, surely, why we don't know is that we only have a qualitative understanding of the wound healing process. And so I should say that most people who are working in the field tend to think that the immune system is a big part of why you have chronic wounds, immune system going wrong in some way. But it's definitely only rather qualitative, and definitely not good enough to start determining therapies or how you could improve on that. Okay, so So in some sense, the question that motivated us, myself and my collaborators who worked on this problem, was really trying to understand better about chronic wounds, why some wounds are chronic and why some are not. That was our big question at the beginning, and we still don't know. We're very far from it, but we've learned a little bit maybe in the last 5 to 7 years or so. Now, typically, it's, as you can imagine, it's quite hard to do systematic experiments on people for various practical and ethical reasons. So what is often done is wounds are studied in what are called model organisms. It's something that happens a lot in biology. And typical ones which people study are at the insect scale. You have fruit flies, you have fish, and then you have mice, which are maybe closest to us. Actually, today, I'm going to tell you about some stuff that we've been doing, looking at wound healing in fruit flies. So I'm not really going to talk about that. But at the end, maybe I'll say something about where I think how that links to maybe wound healing in things like ourselves. And like all research projects, this is usually done by many people. So the work that I want to describe today is a collaboration between mathematicians and biologists with a little bit of inspiration from clinical medical scientists. Keith Harding here had the leading chronic wound clinic in the UK. But of course, like all research, the thing that drives the research are brilliant, hardworking young people. And so the people who really drove this research are the people in blue, and that's Jake and Deems, former students, Mark, former postdoc, and Henry, current student. And this, of course, was done with various colleagues in maths in Bristol and also biochemistry, researchers in biochemistry. So, as I said, the people in blue, whose pictures are circled in blue, those are the heroes of this story, and I'm just a narrator. Okay, so what's the system we start off with? So, this is done by our biological colleagues, who if you want, the biological PIs are Helen and Paul, and and really one of the things that I kind of want to get across here is how much new experimental techniques, many coming from physics, are really revolutionizing how much we know about these kinds of systems. So I'll just say, so the experiments that our colleagues are doing, and I should say that these experiments I'm going to tell you about are done by Jake, the former student, who has a background in mathematics, did an undergraduate in mathematics in Bristol, but then doing his PhD, spent equal amounts of time in the lab, in Helen and Paul's lab, as he did in the maths department in Bristol, and did some amazing work. He's now at the Mechanobiology Institute in Singapore, which is one of the leading institutes in this area as a postdoc. Okay, so there is if you want, this has now become a rather standard technique. You can fluorescently label particular proteins, and Helen is an expert on the genetics of the fruit fly. So what we are doing is the techniques that, if you want, she's developed were to take some proteins in a cell, attach a fluorescent probe to them, which then can be observed in a microscope, and then you can track what these proteins are doing during the wound healing process. And that essentially allows you to start asking very precise questions about what's happening during wound healing. And in principle, you could almost label almost any protein you want, but we're going to label two particular proteins. One protein is called E-cadherin. it's a protein that's found on the boundary of the cells, on the cell membrane. It's associated with adhesion between cells. And histone, which is something which is DNA wraps around. So essentially tells you where DNA is. So essentially, and so what you could say is that you have two dyes, one green, one red, fluorescent dye. So you shine light on this thing, and then it gives off light at a certain frequency, and you can see where things are. So you can see where the membrane of the cell is. You can see the boundaries of the cells, and you can see the cell nuclei. Okay. So, but the other thing that's interesting is, for pragmatic reasons, you have to do your experiments on something, and we're doing experiments on the fruit fly, and we're choosing one phase of the fruit fly, the pupa phase. And the reason why we chose the pupa phase is because in the pupa, it's a quiescent phase, so the animal doesn't move. So it's very easy to image. So that's the reason why you choose the pupa phase. And even more, within the pupa phase, we are going to be looking at the wing of the pupa. And again, as a pragmatic reason, the reason why you choose the wing is because it's two-dimensional, so therefore it's easy to image. Okay? So this is an example of the sort of image that you would generate. And you can sort of see the boundaries of the cells, and you can see the cell nuclei. And this is a side view of the image. And this is done using a technique called confocal microscopy. By the way, if anybody has questions, please feel free to ask at any point. Just raise your hand, and I'll do it. Okay, so essentially, what you can see here is we can sort of resolve individual cells. So that's really obviously very, very interesting. And so we can then perform experiments where we make a wound in the wing, and we can then, of course, follow how the wound heals. So the couple of things I should say is that in order to get reproducible results, it's important to be very careful about what you do. So it turns out that we had to choose a very particular point in the pupa life cycle. So we really choose it 18 hours after the pupa has formed. That's very important. Of course, the wounds will heal other places, but they'll heal differently or in ways which are not quantitatively comparable. So in order to be quantitative, we have to do that. And it also something we learned through hard experience. It's also important where you make the wound. So if you make a wound in one part of the wing and another part of the wing, you get different answers. Again, qualitatively it might be the same, but if you want to be quantitative, then then it matters where. So in some sense, we need to be specific both in space and in time, if you want. So we need a sort of a space-time description of this process. Okay, and I should just say that so in this part of the life cycle of the fruit fly, this is an area where the wing is sort of extending. So the wing is sort of extending. So we're at this point where the wing is going from something which looks rather bunched or something like that. Whilst our wound is closing, this is happening as well. There's growth of the tissue. And that's one of the reasons why it's hard to understand wound healing, because it's not happening in a passive thing, it's happening in a dynamic environment. So you are trying to understand a perturbation of something that's dynamic, and that's hard. So this is just a typical video of the wound healing process. We typically, so what we have is we have many such videos, and so essentially it's the collection of that large amounts of data. This is what will allow us to try to understand what's going on. So in some sense, we've got this new technique which allows us to collect data, but which allows us to study this thing in a great detail. But of course, there's going to be lots of data, lots and lots of data. So one of the problems we have to deal with is, where's the pointer? Okay, there it is. Okay, is the issue of there's large amounts of data. So I guess this is just saying what I just said. So we can actually start asking really, how does the wound healing occurs? And this is something you couldn't say without these types of studies. You couldn't say how it happened. You could only observe it at the end or something. You'd say, okay, I know what this is what it starts off like, and this is what it ends like. But you don't know what's happening in the middle. And now we can say what's happening in the middle, and then we can maybe understand it better. And sometimes that's where, that's really what this new, if you want, frontier of quantitative biology kind of allows you to do. Okay, so I guess it should be hopefully clear to you that we're doing interdisciplinary research here. It's a very complex system. There's chemistry, because of course there's signals from the wound. There's physics, because there's things are moving. And there's maths, because I've got to ask how much. So in some sense, all of us who are in this collaboration have to become jack of all trades. We have to learn things about different areas, maybe move outside our comfort zone. Okay, so that's an important part of the philosophy one will have to take if one wants to do this type of interdisciplinary research. Okay, so now we have this. So I've set the scene for where we are. And so I've got to ask a question, okay? So I've got to ask a question, and then I've got to try to answer the question. So I'm going to ask a very simple question. So the question is, just how does the epithelium heal? Which is, I've already telegraphed it from the previous slide. So the question is, and I can ask this now because I can answer it. Because I can, if I have a model of how it does, then I can test this model because I can observe things. Of course, this thing is a very complicated, complex process. So you can see that asking the question, how does it heal, is not very restrictive, because I could ask about it on many scales. I could ask about it, maybe, is it a chemistry? Is it a chemical thing? I can ask, or is it a physical thing? So in some sense, in order to even to ask this question, I need to start thinking a little bit more precisely about what I mean by how it heals. Okay, so I've got to so how do I decide what I mean by how it heals? So then I have to basically make make that more precise. Let's try to make that a bit more precise. Okay, so now I'm abstracting somewhat. So I have to sort of what's happening is a very complicated biochemical, biophysical process, and I'm going to abstract it into three things. I'm going to call healing processes. Okay, so one process is that the cells can change shape. Another process is they can move, and then the third process is they can divide. Okay? So my question then of how it heals is, now I can make it more precise. I can then say, how do the shape changes of the cells, how does cell motion and how does cell division, how do they contribute to the wound healing process? So this is where I'm going to ask my second question. So I've now turned it into three choices, three things. So I'm going to ask people, and it's a somewhat vague question, so maybe it's a bit unfair, but then it's not really important if you get it right or wrong. So I'm going to ask, so which one of these things do you think is the most important, or what do you think, or what's the relative importance of these processes in this wound healing process? And I'm gonna probably try to do a vote. So I'm going to ask each of those. So I'm going to ask shape, and then I'm going to ask people to lift their hand if you think it's shape, and then I'm going to ask. Okay, so since we're shape, who thinks shape is going to be really important? Nobody. Okay, so shape down. Motion, who thinks motion is important? Okay, I don't know, it's probably about 15 people, something like that. Okay, who thinks cell division is important? Oh, so clearly cell division wins. Okay, so now let's see, because obviously I wouldn't ask it if I didn't have something to say about it. Okay? So what I'm going to do in the rest of the talk is I'm going to really talk about looking at divisions, shape, and motion, and then I'm going to see how much of those, how much they contribute, and or how they contribute. And it'll turn out that it's not just how much, but you have to ask a more complicated question, a more subtle question about exactly how that happens. Okay, so first thing is I should just make a comment about, we have lots of data, and so we put a little bit of mathematics. So of course we all know, hopefully, about the real numbers. The numbers, all positive and negative numbers, including fractions and irrational numbers. Okay, and we call that R. Okay, that's a set of real numbers. And of course we can think of sort of pairs of real numbers. So we can have R1 and R2, two real numbers, or we can think of like 5 real numbers. You have an array of 5 numbers. Or we could have N, where we have an array of N numbers. And essentially, what I mean by big data, or lots and lots of data, is what we have something like this, but N is very, very big. Okay? And so what we have is these videos, remember? These videos of the wound healing process. So this is happening in time. And actually, and of course, what we have essentially some different channels, different colors, and we have also we are also measuring things in time. So we've got various time points. So that's what these numbers mean, basically. And we also have maybe pixels of the image of our video. So essentially, each of our videos essentially has R to the 2 billion. So we have 2 billion numbers for each video. And the point is that lots of these numbers are not useful, but some of them are useful. So what we are trying to do is we have to somehow extract these useful numbers, which I put signal S, from all the non-useful numbers. And when you have a large amount of data, that's not necessarily an easy thing to do. You have to be smart and efficient about it to find ways of getting this information. And so that's the first thing that we will have to do in each of these things. And we are going to need essentially mathematical tools, really just to observe the videos, to analyze them, and eventually, hopefully, to come to some kind of explanation of what we're seeing. Okay, so as I said, the beauty of having this kind of system is that we can just ask any kind of question that you want about what the wound does. So, for example, we can just ask about the area of the wound, and this is another wound, which is just wound, and just watching the wound, and you can sort of see that eventually the wound closes, and I'm going to and what I want you to get from this is that, so I should say the wounds that we talk about here are typically of the order of maybe 10 to maybe 50 microns in diameter. That's a micrometer, which is a millionth of a meter in diameter. So that's kind of how big our wounds are. And the only thing I wanted to kind of tell you is that the hole, which we started with, is gone in less than an hour, because these are the biggest wounds, these are the smallest wounds we have. Okay, so basically the hole is gone in an hour. Okay, that's the thing that you should bear in mind. Okay, but the other thing you should get in mind is that we have a huge amount of information in that, which we're slowly beginning to tease out, but there's loads to do still. Okay, so now I'm gonna get to looking at divisions, because that was the one that everybody said was the most important one. So let's do that first. You see, I had it first, so I already knew that you were going to say divisions. Okay, so the question you're going to ask is, how can we track divisions? Okay, so I'm just going to show you a video here, not a video, some frames of videos, and this is healthy and wounded tissue, and then and sort of seeing what does a cell division event look like. Okay, so you can sort of see you have this cell, looks a bit bigger, it gets big, and then you see the nuclear material splits into two. This is called mitosis with the biologists. And then you see that they split into two, and then eventually you start seeing the formation of of an interface between them, and then finally you've got two daughter cells which have there. Okay, so we can actually see it in the video, and then this is the same thing happening in a wood. And typically what you should see is that this is happening over a time scale of around 10 minutes. Okay, so in order for us to understand what's happening in the process of the wound healing process, we need to be able to track lots and lots of divisions. So we need, thousands and thousands of divisions. Although, if we just look at one division, we don't know whether it's representative. The way we know whether something is representative is if we can make many, many measurements. So we need to be able to do this for thousands and thousands of divisions. So that looks really not very nice. And actually, so initially what happened was Jake would go through the videos and he would look for this, and in the video you annotate the video to say there's a division here at this point. Okay? So poor Jake did this for a few thousand divisions. But we need more. That's not good enough, just a few thousand divisions. Plus, I'm pretty impressed that he did it, actually. Anyway, so we need something better. And, well, it doesn't take, maybe, it probably is not going to surprise you that probably this might be the kind of thing that a neural network might be good at, or in other words, you could say machine learning would be good. And of course, I must say, we did this. This is stuff that we've been doing over the last 5 years. So we did this well before the Nobel Prizes were awarded. We started thinking about these things, though it was probably not surprising. You didn't need to be a genius to figure out that Nobel Prizes were coming. Okay, so in fact, we're going to use machine learning to actually collect the data. So, and we're going to use a type of machine learning called deep learning. So machine learning essentially is, we're going to use it for essentially something which it can be used for many things, but for us, we want to use it to classify objects, so that's a classification problem. So what you want to do is classify is information in some way, and deep learning, and so you have a neural network, and typically if you want to I think there's two things to remember. One is you have a set of nodes connected by links, and so the two things you need to worry about are how do you vary how are the nodes connect to each other, and how strong are the links between them. And typically what you do is you set up with some architecture, so some set of some connectivity matrix between these nodes and links, and then that's fixed. And then what you vary is the strength of these links, and then this is what's called learning process, and then you optimize it in some way to learn something. And that's what we're going to do. We're going to do that we want to do that now to figure out these cell divisions, and we're going to use a type of neural net or deep neural net, which is used in computer vision, and typically it's something that's used to segment images into categories. Okay, so typically imagine that you have an image like this, a picture, and this picture has bits of sky, tree, building, road, cars, and road markings, and then so you've got an image. So you've got an image, so this has a certain number of pixels in this way and this way. And you can sort of say, roughly speaking, your number of nodes is related to your number of pixels. So in the end, you still want to have the same number of pixels. But what you want is to be able to take bits of the image and say this bit of the image is sky, This bit is tree, this bit is building, and this bit is car, this bit is road. So it's called a U-net because you start off with a certain number of nodes. You go to a small number of nodes in your layers, and then at the final you come back and you have the same number of nodes you started off with. So the network has this sort of structure where it has lots of nodes at both ends and a few nodes in the middle. And this allows you to classify things. We're going to use this, but we want to use it now not for images, static images, but we're going to use it for videos. We need to use it for videos. So really what we want to do is classify our video into some events of finite duration, whatever it is. So what the U-net did was it said, okay, the traditional U-net said, well, I can specify which class each pixel belongs to. Now I've got essentially a 3D object. I've got my two space dimensions, and I've got time. Okay, but I still, again, can create a sort of U-net architecture which can take into account this third dimension time. And then we can now identify any type of event we want to. So to cut a long story short, we did this so that we can now automate the collection of divisions. And so now I've got divisions here. I'm going to play the video now. I'm going to play it a few times. I'm going to ask you some questions. And the question you'll see divisions, where the divisions happen is going to be a circle, and there'll be a line through a diameter of the circle, and that's going to tell us the plane of the division. I'm just telling you what it is. I'm not really going to talk about the plane of division, but if you want to, I can talk with you about it later. Okay, so this is now the video, and now I've shown you where the divisions are happening. I'm going to play it a few times, and I just want you to tell me. Now I'm going to ask the audience to tell me what you see. What have you seen? Okay, do it. Oops. Okay, I think this now I have to do it by hand. Okay, do it again. I'll do it the third time, and then I'm going to ask for input from the audience. One more time. Okay, so did you notice anything about the divisions? Anything?
It's a build-up of patterns in certain areas, and they resemble. They move around to space.
Okay, so I'm going to summarize what I think you're saying. You're saying that it wasn't uniform. It was heterogeneous. There was somehow variations in divisions. It's not uniform every space. There are places where there are more divisions and places where there are less divisions. I think that's okay. Any other observation?
Because last divisions found that the number of divisions increased throughout the middle of the year and then decreased again.
Okay. Wow. That's the kind of person I want. Are you doing a PhD? You want to do a PhD in Bristol? Yes?
Repeat what was said.
Okay, so I'll repeat what was said. She said that there seemed to be less divisions at the beginning. There was a peak, and then it went down again. Okay? Anybody else? Anybody else noticed anything?
They're not happening in the womb.
Good. So not only were they not spatial, the point you made, but there don't seem to be any divisions at the wound. Okay. So first that's videos. Okay. One more.
My observation also was that the mean distance from the wound was kind of like constant in the circular.
Okay. Woo. This is really a very, very perceptive audience. So, okay, good. So saying that somehow it seems the divisions were happening at some fixed simply localized at some fixed distance from the wound. I think that's the point you're making. Okay, good. So now the point I just want to make is that the eye is a great guide, but maths is even better. So of course we have all the information. So I can actually now, everything the people mentioned was right. Now I'm going to show you it's right. Okay, so just to kind of so this is of course so now you can sort of see that the divisions. So I should say that so first we're going to look at unwounded tissue just give us a baseline, and we want to compare the wounds to that. And remember that this is this pupa phase where the thing is decaying towards its adult. So therefore the number of divisions is actually going down because you have more divisions when you're young, and as you get older you have less divisions. So this is sort of, so there's a sort of a trend which we know, which is that. And now we put in the wound, and this is just time. And as somebody brilliantly noticed, you see that when you have the wound, there's this dip in divisions after the wound, and there's a burst of divisions, and then it delays back to the background level of divisions. Okay? The thing, though, I want you to point out is that if you noticed, remember the wound closed about in less than an hour. Okay? So here, this is when the wound is closing. No divisions. But after the wound has closed, there's a burst of divisions. Okay? So clearly, divisions plays a role, but it plays a role, it's not a temporarily uniform role. It's something, it really happens later. It happens after the hole has closed, but probably it's helping to heal the tissue. And then this is now, we can do statistics we can also look at what's happening as a function of distance from, we can just do a space-time map of the divisions around the wound. And again, you can sort of see here that indeed there is, we can see the time. This is saying there's a burst of division time, but there's also a spatial correlation. The divisions are happening at some distance from the wound, and in fact, we can do a little bit more than that. We can show that this distance scales with the size of the wound. So if you have a bigger wound, this thing happens at a bigger distance. Okay, so therefore, you know, the division, you know, is maybe not as important as you thought it was, but it is still important. Okay. Okay. So this is just summarizing what I said. And actually, nobody mentioned this, but if you looked at the planes of divisions, there was no correlation with the wound. You could have maybe, if you looked at the video a bit, you could have seen that as well. But that's not something I want to talk much about. Okay. So I'm gonna say now briefly some things about shape and motion. Okay, so let's do shape. So again, you know, one of the beauty of having this sort of lots of data is you can really analyze it, right? So you could turn your image of the cells into essentially you can think of the cell boundaries as a bunch of polygons. Okay, so essentially your cells are a bunch of polygons, and then there's a way one can quite systematically map each polygon to an ellipse. And if you want, I can tell you how to do it, but afterwards. But roughly speaking, each cell I can turn into, and I can map to an ellipse. And this ellipse, of course, will have two parameters corresponding to the major and minor axes of the ellipse. And essentially, if these two numbers are, of course, roughly the same, then I get something which is mostly round, and if they are different, then I get something which is more elongated. Okay, and of course, the axis of elongation, of course, is also property of the cell. So one thing is that if you want, we have a way to go from those images to a bunch of ellipses, and so we can track so we can track what the shapes of the cells are doing. And more or less, I can summarize what you see in the following, that we can look at the shapes of the cells close to the wounds and shapes of the cells far from the wounds. And roughly, this is what we see. So far from the wounds, there's an axis, the cells tend to be elongated and oriented in a particular direction. And this direction is this direction of the wing growing, actually. That's what it is. But close to the wound, the cells are, in fact, more elongated than there. But they tend to be, in general, elongated, but aligned along the edge of the wound. And this happens immediately after the wound has happened, literally as fast as we can image. So it happens very quickly. Okay? So in fact, the shape is the thing that reacts quickest to the wound. Okay? So we can actually, if you want, you can then sort of go from this, if you have those two conditions, far away, it's parallel, then in fact, it's just a question, it's just a topological statement that in fact, this must, essentially, this modulo much more complicated structures is what the pattern of shapes must do. Okay? And this is related to topological defects. I'm not going to say anything about it, but just there's interesting mathematics in even looking at that. Okay? So, but the point about it is that, so once now I've got the shapes, so I can now define the directive, I can define a vector, which is the major axis of each of the shapes. Okay? And it's not really a vector. It's actually a second rank symmetric traceless tensor, but you can think about it as a vector which is headless, so that pointing this way is the same as pointing that way. So this is basically, so this is a map of the shape of the cells near the wounds. And then once I've got these major axes, I can just get rid of the cells. And so I have now a set of these vectors at every point in time. And remember, this is the initial condition. This is what happens immediately. So now so this is now essentially a field of directors n. So that there's an n now, a director, which is my shape of my cell, at every position in space and at every position in time. So we can now ask, what do the cells do? Okay, now we can solve this. So and the way we do that is so now this gets a little bit mathematical. So I can think of now my shape is a vector field. So it's a vector, a smooth function. How smooth it is, one has to be a bit careful about defining how smooth it is. Up to some point, smooth function, which varies in space, around, and in time. Okay? And then I have motion of the cells will be correspond to another field, which is the velocity of the cells, which again can vary in space and time. And so if we want, what we want to do is to map the dynamics of the cells to some equations of motions for these fields. This director field, telling local elongation of cells, and motion. And one can write down such equations. They involve forces, chemical reactions, and something which are called active couplings, which couple mechanics and chemistry, and they also have fluctuations. And to understand this, you need to use a field which is called active matter, which is essentially my main area of research, I would say, over the last maybe couple of decades, and non-equilibrium statistical mechanics. So to understand that, you need to include those bits. But anyway, we can put those things into. So what we want to do is to come up with some set of equations of this thing which matches the experimental data. I won't go into details how you do that. You can do that by comparing observables of the data with parameters in the equations. And so now we can now look at the evolution of this director field and this velocity field. Okay, and this is sort of what's happening, and I think I have some videos, and so if you want, and so you can, so this is the seeing the, and those points there are what are called, these are called topological defects, and they relate to, they're part of the, they are required just by having those boundary conditions. Okay, so in some sense, what we see is that and what's happening now is that the cells are moving to close the wound. Okay. So what have we learned? First thing, first that the three things all contribute to the wound healing process. But actually, and this is something that surprised us. I had thought we would just see them working in parallel. Actually, what happens is they work in series. The thing that happens first is the shape changes happen first. So the cells elongate along the edge of the wound. That's the first thing that happens. And then motion happens next. The cells move in to fill the hole. And then finally, the cell divisions happen last. Cell divisions are the last thing that happens. And so in fact, in terms of modeling, it's actually nice because it means that you can model them independently because there shouldn't be much cross correlation with them. So it's a serendipitous thing about the things. And so you could ask why. Now that's a topic of a different talk. So I'm not going to tell you, I'm not going to say anything about that. And probably also because we have some ideas of what we think is happening, but I wouldn't be willing to go state them in public. Okay, so so in any case, so that's the thing. So things is, where do we go from here? So what do we do next? Okay, so in some sense, what we've done is we've parameterized the dynamics of what I would call a wild type wound healing in this people wing, and it's kind of important to understand that this is very specific to this particular tissue. So some of the ideas will be generalizable, but what the things we know are very specific to this particular system in a sort of mechanistic fashion. But the point is this, now we have a sort of parameterized model of this thing. So we can now use this as a baseline to start applying chemical and physical perturbations and see how they affect the wound healing process. And so if you want, then this is a way you could start thinking about maybe therapies or things like that. I mean, obviously, I should say that the caveat is that just because something works in fruit flies doesn't mean it's going to work in humans. So we should really, that's a very big caveat. But nevertheless, one thing you can say is that there are certain proteins which biologists call evolutionarily conserved. So that means that you find analogs of these proteins in very different organisms, and a lot of the proteins that are involved here they're analogs in humans. So therefore, it's not completely crazy to think that some of these things might be relevant to mammals of one group, but that's definitely a huge caveat. This is some of the papers that describe some of this work if you want to know more about it. And I think I want to finish with my thing I started off with when I talked about when I said that we have to be jack of all trades. And I found this very interesting to look at the history of the phrase jack of all trades. So I did some sleuthing, not very detailed, mostly on the internet. And I wanted to look at, essentially, I wanted to do the time evolution of interdisciplinarity, And of course, interdisciplinarity means you're a jack of all trades, okay? That's what it is. And the earliest one I could find was a 1592 attributed to Robert Greene, but maybe not Robert Greene, about Will Shakespeare. And he talked about this actor who had the temerity to write plays. And he said it is a Johannes Factotum, which means jack of all trades. And so that was the word, jack of all trades. But of course, many of us have heard a longer version. What's the longer version that people have heard? Master of None. So when do you think Master of None appears? Well, I don't know. Again, these are vague, and I'm certainly no literary study, so around 150 years later. So there are a number of cases around the 1720s where you see jack of all trades, master of none. Okay, so that says that's not a very positive view on interdisciplinarity or doing things. And this continues, more or less this sort of thing continues for the next 250 years. And then around 2000, so I really want to say that these statements are not fixed in time, they vary. So around 2000, you start seeing people, this bit gets added. And the earliest one I could find was Principal Magazine, which is the National Association of Elementary School Principals. And essentially, it's sort of interesting because sociologically, this is when our moment in time is a time when interdisciplinarity is clearly going to be very important. We need to get teams of people with different skills to work together to solve big, complex problems. So it sort of says that, sayings aren't fixed in time, they evolve, and they somehow reflect the time that which they exist. So I think I'll stop there. These are the people who did the work. Thanks.